In this video, we're going to be talking about the biological stains and tests that you have to know for the USOO. So let's start off with the Bure. So this is for protein slash peptide bonds. And what happens is when you have unreacted proteins, that's going to give you a deep blue color, which is this one. And then when you have peptide bonds, this is going to give you a purplish color. Now for Benedict, this is going to be for carbohydrates. And then when you have an unreacted carbohydrate, that's going to give you a turquoise color. But when you have a reducing sugar, like glucose, is going to be yellow-orange. So can you remember sugar and like heat is going to give you warm colors because it's going to be heat, like think warm colors. Now for non-reducing sugars like sucrose, that's going to be green. And like for the green, you can think of like sucrose is like, kind of think of like sucrose with the color green. I don't know why. But yeah, so yeah, unreacted turquoise, just think of like a hospital. Like a hospital, you're wearing like blue gowns and you're like unconscious when you're wearing those gowns, so it's blue. Okay, now for ninahydrine, that's gonna be for amines and amino acids, and amino acids, they're gonna be a bluish purple, and just remember that. And then for proline, you just have to remember that proline is gonna be the odd one out, and then from the other one, you can determine that amino acids is blue purple from using the process of elimination. So proline, this is your special one, it's the special case, which is the yellow one. And proline is almost like it's similar to the spelling of urine, and urine is like yellow, so you can kind of think of like that. Now for iodine slash Lugol's, this is gonna be for starch, and when you get a positive result, this is when you get when you have unreacted slash amylopectin slash other types of carbohydrates, that's gonna give you yellow orange color. But so that's gonna give you your yellow orange color right here. And then when you have a starch, which is like amylose, that's gonna give you a blue black. So just remember this. Starch stains dark. Starch stains dark. Starch stains dark. Okay, now for Sudan. This is going to be for lipids, right? So now lipids, lipids are oily, right? So with oil, that water and oil don't mix, right? So then you can think of like how there's going to be fat floating, and there's also going to be a stain. It's going to be yellow-orange, and the fatty part is just going to be orange, and that's just going to be the color. Okay, so that's it. So just repeat in your head over and over and over and over and over and over and over again all of these things until you remember them. So remember some traps that you want to be aware of is that Benedict is not going to be equal to starch, and Bure is not amino acids, that's ninahydrine. Purple is not always going to be purple. I mean, purple is not always going to be protein, because sometimes you can have unreacted proteins, remember? And sucrose is non-reducing, is going to give you a weak slash green Benedict result. And proline breaks the ninahydrine rule, okay? So what is it? Proline is yellow, okay? So remember, when you see a color, just ask what molecule causes that color in that test, right? So again, Bure, purple, protein. Benedict, sugar, warm colors. Ninahydrine, amino acids, purple. And then proline is going to be yellow. Iodine, think starch, is going to be blue-black. And Sudan, think lipids, is going to be yellow-orange because of oily stuff, okay? So good luck.